Good day, this is Professor Wills, CBMG PhD. It's today's Friday, January 14th, 2022. It is 1:45 and 22 seconds, a.m. Eastern Standard Time. Today's research, colorectal cancer. So let's hop to it here. The mouse study identifies bacterial protein associated with colorectal cancers. Newly identified toxin, common in E. coli, bacteria-accelerated colon cancer, in study mice, confirming link to human colorectal cancers, can pave the way for developing preventive drugs. Date January 12, 2022. Source, Johns Hopkins University, Bloomingburg School of Public Health. The strains of bacteria that cause common food poisoning symptoms often contain a toxin that can damage DNA in intestinal cells, potentially triggering colon cancer. The discovery raises a possibility that some of the roughly 2 million new cases of colorectal cancer every year around the world originate from brief and seemingly mild food poisoning events. It also points to the possibility of future drugs that prevent colorectal cancers by neutralizing the newly identified toxin, USH-A. Prior research has suggested that certain bacteria that reside in the gut can trigger colorectal cancer via persistent infections involving chronic gut inflammation. The short-term infections causing food poisoning, including traveler's diarrhea, which normally resolve in a day or two, have traditionally been considered non-carcinogenic. Researchers hope this study will motivate other researchers to do epidemiological studies to investigate this potential link between transient diarrhea infections and colon cancer development. The researchers performed experiments with a mouse model of transient bacterial diarrheal disease using the bacterium Citrobacter rotatum, which has strong similarities to diarrhea-causing strains of the human-infecting microbe Escherichia coli. The researchers observed that Citrobacter infection quickly led to strong signs of DNA damage in gut lining cells in the mice. The scientists also noted that the damage was dependent on a mechanism in the bacteria called the type 3 secretion system, and this syringe-like appendage is used by some bacteria, including Citrobacter and diarrhea-causing strains of E. coli, and to be able to inject proteins into host cells. This mechanism facilitates the invading microbes' growth and survival. The researchers eventually zeroed in on the T3SS injected protein, USH-A, that accounts for the DNA damage. They found that USH-A, which can also be produced by diarrhea-causing E. coli, contains a short segment with DNA-breaking enzyme activity. The function of this DNA-breaking element in the life cycle of Citrobacter is still unclear. In the study, deleting it didn't seem to impair bacterial growth or survival, but the researchers found evidence in their mouse study that USH-A can have a definite cancer-causing effect on the infected host. The scientists experimented with a genetically engineered line of mice that spontaneously developed colon tumors, and found that infecting these mice with USH-A containing Citrobacter dramatically accelerated tumor development. The researchers also found that the types of mutations in the Citrobacter accelerated colon tumors were highly similar for those that have been cataloged in human colon tumors, again underscoring the potential relevance to human health. A strong confirmation of this relevance won't be easy to accomplish, since transient infections, by definition, would be long gone by the time tumors develop. Colon tumors typically develop for many years before they are detected. Researchers say the studies might best be done in sub-Saharan Africa, where diarrhea-causing bacteria infections and colorectal cancers are relatively common. In principle, you can give such inhibitors to patients who present with diarrheal disease to protect them from cancer-promoting DNA damage. Good day, this is Professor Wills, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.